Protein synthesis is the process of how cells make proteins. DNA is 4 different base types: adenine and guanine, which are purines, and thymine and cytosine, which are pyrimidines. Protein synthesis can occur in two ways: transversion or translation. Transversion is when a pyrimidine is swapped for a purine or vice versa. Translation is when a purine is swapped for another purine or a pyrimidine is swapped for another pyrimidine. Here is our DNA coding strand. Note that the coding strand is in the 5' to 3' order and is read from left to right. On Halloween, you meet a witch who knows how to create pumpkins with magical powers. The witch gives you special instructions to make the pumpkin. The first step in her instructions state you must change your DNA sequence to RNA. To do this, we must change our thymine bases to uracil bases. The witch then gives you a key to interpret your mRNA strand. The goal is to group the bases in groups of 3 that match up with the codon table. The witch tells us the hint that our start codon will be the letters AUG. By looking at the table, AUG codes for the amino acid abbreviation MET. The next matches we find are GUC, AGC, and AAG. The next group is UAA, which is a stop codon, meaning that we are done with coding our RNA sequence. The other key the witch gave to us tells us how to carve our pumpkin to give it magical powers. By matching the amino acids to the characteristics, we will carve a smile, eyes, nose, and 3 teeth on our pumpkin for it to have magical powers. Your brother sees your pumpkin and wants to make one just like it. He writes down the DNA sequence for the pumpkin and carved his own, but his finished pumpkin has 2 teeth instead of 3. Where did he go wrong? By looking at the sequence, we can see that your brother copied down a G instead of an A. While your letters AGC code for the amino acid ser, his letters GGC code for a different amino acid gly. The change from A to G is a translation since we are not changing the base type. Because we ended up with a different amino acid, this point mutation is known as a missense. Because it coded differently, his pumpkin will have 2 teeth. Your friend wanted to make a magical pumpkin, so he quickly copied your sequence down but made a mistake. You find that your friend copied down a G instead of a C, which is a transversion. This changed the coding from a GUC to a GUG, so why do your pumpkins still look the same? Both sequences code for the same protein, Val. Because they code for the same protein, you will carve the same thing on your pumpkin. This mutation in the sequence is a silent mutation, meaning that although there was a point mutation, the polypeptide sequence remains the same. Your cousin comes over to see your magical pumpkin that you created and wants to make one of her own. However, she also makes a mistake. She wrote down a T in place of an A, which is a transversion. When this was changed to an RNA sequence, it read UAG, which codes for a stop codon. Because the stop codon in the sequence comes so early, there is not enough amino acids, and this is known as a nonsense mutation. Therefore, her pumpkins will have eyes, a smile, and a nose, but will have no teeth. Your neighbor also wants to create one. They quickly jot down your DNA sequence. They are surprised to find out their pumpkin looks nothing like yours. Apparently, they wrote down a T instead of an A, which is another transversion. When transcribed into RNA, it became UAU. Because of this, your neighbor's sequence contained no stop codon, so they continued to carve and carve until there was no more pumpkin.